Good morning, everyone. For those that don't know you, I'm Beryl, the Director of Tern, and I'm not welcoming you, I'm just giving housekeeping at the moment. So if we have an emergency, we have decided that you're all going to follow Peter Brady, and he is standing next to the door, so memorise that face. But if you can't find him, go out the door and turn right and head for the stairs that are there. That's basically it. And in roughly the same direction, you'll find the toilets, which are out there. We have quite a few people who have had cancelled planes and will be drifting in from time to time. So please be kind to them. They're not really disturbing you. They're just trying to get here. And for people who are online, who have had to stay away because they've caught something, sorry about your illness, but thank you for joining us online anyway. That's really good. If you do want to go out during the session, can you please go up the stairs to the back and go out those doors? And obviously, if you're using those doors for an emergency exit, you have a little bit more trouble finding your way. We are recording everything, so please make sure it's not your phone that we're recording on the recording. So keep those turned off. And we have the break area is out the door and around the corner, as is the area where we're going to be displaying our exhibits. Thank you very much, exhibitors, and our posters. Now, we found this morning that it's too windy to put our posters up yet, so apologies to people. We will get them up as soon as the wind drops a little bit, and they'll be up for the duration of the symposium, but the poster presentations are tomorrow. And we do have some members of some VIPs, but I'm going to leave those to Andrew. So what I'll do now is introduce you to the patron of Tern Symposium. And we chose our patron, this is the second symposium that we've had Professor Andrew Campbell as our patron. He has a 3 symposium gig, so he'll be around in 2027. I just want to tell you what a fabulous person he is and why he is our patron. And I have to use my notes because his experiences in life are just so varied. So Andrew was the inaugural chair of the Tern Advisory Board. So I remember that first board meeting in April 2010. And Andrew did a fabulous job until he stepped down to take his next role, which was to take over the ACR. And some of you know ACR by title. It is the Australian Centre for International Agricultural Research. And Andrew kept in that role all the way through until 2023. He is managing director of a consulting company called Triple Helix at the moment. But for all his life, he's had influential roles in sustainable agriculture research management. And what I like best is he understands long-term monitoring. So he, where am I? There's such a long list. I'm going to miss things, Andrew, because I can't read all the dot points under these lights. He's written widely on land care and sustainability, and he was the Executive Director of Land and Water Australia from 2000 to 2007. And, this is the really interesting part, was instrumental in the development of land care in Australia as the first national land care facility from 1989 to 92. And Andrew, didn't you just tell me that you're patron for Landcare Victoria? Yes. See, he's not just patron of our symposium. And he has professional training in forest science and agricultural knowledge systems from the University of Melbourne, and I can never say the Netherlands, Wageningen Agricultural University. I know it's the best one in the world, but I get stuck after that one. And he was elected as a Fellow of the Australian Academy of Technology and Engineering in 2018. So he's also a board Director of AgriFutures Australia, a Professorial Fellow in the ANU Fenner School, Principal Fellow in the Faculty of Science at the University of Melbourne. Does ANU and University of Melbourne mind you having dual roles? No, good. Everyone wants a piece of you, Andrew. Yes, I've done patron of Landcare in Victoria, and a former board member of the Peter Cullen Trust and the Future Farm Industries CRC, so knows all about water. And his family's been farming in Western Victoria since the 1860s. And Andrew's been managing the family farm, which is about farm forestry, prime lambs and wool, since 1987, but with the help of a neighbour, because he's off doing all these other things. Please welcome Professor Andrew Campbell, patron of Tern.
Thank you very much, Beryl. Can I begin by acknowledging the traditional owners of the land on which we meet today, the Turrbal and Yagara peoples. I pay my respects to their elders and extend that respect to all Aboriginal and Torres Strait Islander people here today and online. Welcome everybody here and anyone online, and welcome to those members of the Advisory Board and the Science Advisory Committee who have managed to get here this morning, and special mention of Professor Paul Bonington, Pro Vice Chancellor Research Infrastructure at UQ, who serves on the Tern Advisory Board. As Beryl mentioned, I had 7 years at Land and Water Australia, and one of my board members was the late, great Professor Peter Cullen. And at one board meeting, he reminded us, he said, well, why would taxpayers invest in R&D? What's the business case for us spending public money? He said, essentially, we need to create new knowledge. We need to support innovation to turn that knowledge into products and services and impact. And we need to learn as we go along so that at least we're making new mistakes. And if I think about those three rationales for public investment, Tern ticks all of those boxes. And I'll just pause for a moment, parenthetically. Another member of that board of Land and Water Australia was Leith Booley, and I'm very sad to say that in recent days we've lost Leith. She's been a leader in water management and science and governance, particularly in the Murray-Darling Basin as the first chair of the Community Advisory Council, but in many other roles, chair of Sunwater here in Queensland. A great leadership development person through the Peter Cullen Trust and the Rural Leadership Program, and so many other contributions. So a very sad loss in recent days. But when I think back to the early days of Tern, and against those three things of informing research for new knowledge, for innovation, and in particular to be a keeper of the long view so that we do learn over time, there are some other dimensions to the rationale for Tern. We're here on this ancient, distinctive continent, highly variable climate, extraordinary degrees of endemism, where we've learned through bitter experience that imported solutions don't travel all that well when you try and apply them in Australia. So we need to work out uniquely Australian ways of looking after our ecosystems and managing those ecosystems and deriving livelihoods from those ecosystems. And that means we need to be tracking impact ourselves and doing that as efficiently as possible. So if you overlay against that unique ancient continent, high degrees of endemism, and then you plonk a federal structure on it, there's a lot of risk. The Well, they're not apocryphal, the railway line issues. They're actually very real. Having systems in different jurisdictions that don't talk to each other well and don't aggregate well is a major problem. And the fact is, we simply don't have enough resources to be able to afford to do things inefficiently. So the business case for NCRIS, and I love that acronym, I think it's one of the better conceived federal programs that I've seen in my career. National, collaborative, research infrastructure, facilities. So every one of those words is really important in the design of NCRIS. And so we were delighted that we were able to get natural ecosystems, biodiversity conservation, management of natural resources up as one of the initial NCRIS facilities, competing against synchrotrons and square kilometre arrays and research ships, really exciting stuff that politicians can cut ribbons on and stand in front of and bang champagne glasses on. We were trying to defend much more distributed, much harder to get your head around, plots and transects and arrays and sensors and acoustics and satellites and models and all much more complicated things, none of which seemed as sexy as a bloody big new ice-breaking ship or something that can see into space or whatever. So I think it was tremendous work from people here at UQ and elsewhere right across the country to make the business case for Tern in the early days. And we're going to get an insight into those early days with a short video presentation. And Beryl's asked me to remind you that the first part of the video was actually made in 2010. So it reflects the thinking at the time, and it's a little time capsule on where we were at that time. And the great thing about this event is that we're here 15 years on. Firstly, that we're still here. Secondly, that we've got a chance to reflect on that journey and to share lessons and insights from that journey. And it's always good to bring a community together to not just share those insights and so on, but also to think about where to next. And so I'm looking forward to that very much over the next couple of days. So as I said, it's terrific for us all to be gathered here, and it's my great pleasure now to introduce Professor Emily Nicholson, who's our keynote presenter. Emily's got a glittering academic resume. She's the Professor in Conservation Biology in the School of Agriculture, Food and Ecosystem Sciences at the University of Melbourne. She's a conservation scientist who has influenced conservation policy and practice at global and local levels. And her research interests, obviously very relevant to us here today, include biodiversity monitoring and risk assessment, conservation decision-making and ecosystem science. She co-leads the IUCN Red List of Ecosystems thematic group in the Commission on Ecosystem Management and is a member of the ad hoc technical expert group on indicators for the Kunming-Montreal Global Biodiversity Framework under the UN Biological Diversity Convention. She has more than 100 publications, including in Nature and Science, and has been cited very many times and has done extremely well in attracting research grants, including an ARC Future Fellowship, 2 Discovery grants and 4 Linkage grants. So, delighted to welcome you, Emily, and look forward to this very much. Thank you.
Hi everyone. Sorry, it's very bright lights here. Thanks for the opportunity to speak with you today. So today I'm going to talk about some work that's been occupying me and most of my thoughts for the last not most of them, but most of my work thoughts for the last couple of years, which has been working with the UN Convention on Biological Diversity on their monitoring framework and measuring progress towards the Kunming-Montreal Global Biodiversity Framework, and then how that fits into an Australian context. And I'll use some acronyms, I apologise. The main one is GBF instead of Kunming-Montreal Global Biodiversity Framework for obvious reasons. And first I wanted to acknowledge that we're on the lands of the Turrbal and Yagara people and pay respects to their elders and ancestors and their stewardship of this country, and also of the Wurundjeri Woiwurrung people where most of my work takes place and where I live, work and play. Also of the Bunurong and Gunai Kurnai people for this, where I took this photo, which is I guess most of us here would have been inspired by a happy place where they spent a lot of time in the natural environment that really inspired a lot of the curiosity about nature, which is why we do what we do. And so that's near Wilson's Prom, which is a pretty special part of the world. And then finally, just in the context of what I'm going to talk about today, I just want to particularly acknowledge the extraordinary work of the Indigenous people and local community representatives who worked through the negotiations of the GBF to ensure that Indigenous people and local communities were really deeply embedded throughout the goals and targets and indicators. And this is a huge advance even over the UN Sustainable Development Goals, which came a bit over about a decade earlier, which had very limited, pretty much no reference to Indigenous people as knowledge holders, rights holders. They're only mentioned in the I think preamble to the SDGs, whereas in the GBF they're right through many of the targets and in a meaningful way. And in particular, they had a working group that developed indicators of Indigenous people and traditional knowledge, which have been picked up as headline indicators, which means that countries should report on those. And I think that's extraordinary and very impressive work. They also got a new subsidiary body that's set up to guide the CBD in their work. So just as a quick introduction, hopefully I suspect many of you are familiar with the GBF and with the Convention on Biological Diversity, but the Kunming-Montreal Global Biodiversity Framework was the latest set of these decadal targets and goals agreed by countries or by parties to the UN Convention on Biological Diversity. The convention was established in 1992 as one of the three Rio Environment Conventions, along with the Climate Change Convention, which is much better known, with the decadal goals like the Kyoto Agreement and the Paris Agreement, or Kyoto Protocol and the Paris Agreement. So it's the primary multilateral agreement on nature. There's 196 parties, including Australia, so that's 195 countries and the EU. And so the GBF, as I said, was this latest set of goals that was agreed in 2022, has four outcome-oriented goals, which relate to the objectives of the convention: sustaining biodiversity, sustaining the benefits that people get from nature and sharing the benefits. And then 23 targets that are needed to achieve those goals. So it's built around a theory of change, which is a step forward for framing the goals and targets. And then another advance was to have an agreed monitoring framework so that we could measure progress towards these targets and goals, but also that this monitoring framework would help countries implement, so understand where, for example, biodiversity and its values were. And that included a set of 26, I think it is, headline indicators that were agreed by parties and that they would all report on, which was a miracle in itself that you could get all those countries to agree on a set of indicators. And actually, given that these are politically negotiated, they're actually quite good in terms of their roundness and completeness for monitoring. One of the other advances, I think, of the GBF over previous agreements is it took a really strong ecosystem approach. So, for example, Goal A starts with a clause about improving the trajectory of ecosystems, natural ecosystems, in terms of their area and integrity. And then Goal B is about maintaining the benefits that people get from it in natural ecosystems. And there's a whole range of targets that relate to ecosystems as well, such as restoration and ecologically representative protected areas. And we also have this set of ecosystem-related headline indicators. Now, together, if they Is that 5 minutes in or 5 minutes left? Left. Yeah, good, okay, thanks. God, I'm going really slowly here. It's a slight tendency anyway. So this set of indicators have the real potential to be able to tell us a really strong story about how ecosystems are faring if it's done consistently by indicator and if they're reported consistently by indicator and by country. So for example, the red list of ecosystems tells us how threatened our ecosystems are. The extent of natural ecosystems tells us how much is left and if that's going up or down. Service B1 tells us about the benefits we're getting and whether that's increasing or decreasing and where. Area under restoration tells us how much we're restoring, for example, of mangroves. And 3.1 tells us how much is protected of each of our ecosystems. Now, we only get the benefit of that set if we report consistently, for example, by ecosystem type. So we can report this data by for seagrass, mangroves, coral reefs separately rather than lumping them together. And the expert group on indicators proposed that we use the global ecosystem typology for that, which was a new ecosystem typology developed by IUCN, published in 2022. And I admit I'm a co-author on that, so it was not a conflict of interest, but I knew it very well, put it that way. And so this is able to support consistent global reporting, not by replacing national data sets on ecosystems or state level that we have in Australia, for example. Those are really the foundation of action to have locally relevant, locally embedded ecosystem maps and knowledge. But rather it's this hierarchical broad categories of ecosystems to which all ecosystems can fit in. So here's the hierarchy. In red here we might have our national ecosystem types and you can align them with these broad categories, and I'll show you some examples of those in a moment. It can also support the development of new ecosystem classifications. So it was developed by over 100 scientists who are involved in it. It was published in Nature. There's a really great website I encourage you to have a look at. It was adopted by the UN Statistics Division as one of the international family of statistical classifications, along with different job types and different bean categories, agricultural produce. So it's a very formalised statistical classification now with broad acceptance. And so there's 110 ecosystem functional groups, which include anthropogenic ones. It should be all-encompassing across all different realms. And here's just some examples. They're very familiar categories that most scientists would recognise: the tropical forests, coral reefs, permanent lowland rivers. As I said, there's a website, so you've got a one-pager on each of them, indicative maps of where they're likely to occur, and a conceptual model that tells you something about what makes that ecosystem tick, what are the key drivers and the characteristics of that ecosystem. And just to give an example of what we mean by these set of ecosystem groups that occur globally, so it's different from ecoregions that, you're talking about a small part of the world. Here we're looking at ecosystems that function similarly. So for example, tropical, subtropical pyrex savannas, they occur around the world in the subtropical, tropical realm. And here we have three examples from Brazil, from Africa, and from Australia, from Tiwi Islands. And they have pretty much all completely different species, except maybe kangaroo grass that occurs across them. But they function in the same way, where there's this real driver of rainfall, then grass growth, curing, and burning. And that's the key driver of that system that makes it unique. And it might have varying levels of trees, but they're not the key feature of that system, the key driver. They play second fiddle to the grass and the fire. Okay, so how do these indicators work together? So here's an example from Colombia that I developed using their data from their Red List of Ecosystems assessment. So we have the Red List of Ecosystems, 3 headline indicators: Red List of Ecosystems, extent of natural ecosystems, and 3.1 on protected areas. Each row is a different ecosystem functional group. And so we have 80-something, I think it's 80 or 81 ecosystem types across these, 10 or so ecosystem functional groups. And when you put these together like this, suddenly you can start to tell a story about how these ecosystems are faring. So for example, dry tropical forests, we can see all red. That means they're all critically endangered. Why? We can see that a big driver of this has been the loss of area. There's virtually nothing left, something like 5 or 6% left of the original distribution. it's so little left that you can barely see in the protection indicator, but about half of what's left is protected. And some of that was new protected areas that were established when they did the Red List of Ecosystems and realised just how threatened these ecosystems were. They're also home to critically endangered species like this cotton-top tamarin. And then we can compare that, for example, with tropical lowland rainforests. This is the Colombian Amazon. Contributes this. About half of these are threatened, which is pretty concerning. Less of a loss of area. So there's other degradation that's going on that's driving this risk that they're threatened. And they've got reasonably good levels of protection, nearly up to 30%, which is the target in terms of area under the GBF. So I'm just going to dig into a couple of the indicators for ecosystems. So there's the first one is the Red List of Ecosystems. So again, it's something that I've been involved in developing and doing research on for the last 15 years with David Keith and Jess Rowland was a PhD student of mine who worked on how to turn the Red List of Ecosystems protocol into an indicator, which was then picked up by the GBF, which is pretty amazing for a PhD project. So a Red List of Ecosystems is about knowledge synthesis. You're bringing together all the known knowledge about that ecosystem type, descriptions of the characteristics of that ecosystem, maps, data on change in area and integrity or degradation, and diagnosing the threats, the key threatening processes for that ecosystem. We then assess against a formalised set of criteria with quantitative thresholds over different timeframes that relate to change in area and in integrity or degradation. And then through this, we can allocate them to a risk category. And this is a map from South Africa's National Red List Assessment, where they map their threatened ecosystems, and they found that to be a really powerful tool for mainstreaming. And then from those risk categories, we can develop a Red List index. So they were the dots in the Colombia page, where we've just got one assessment. In South Africa, they've got multiple assessments. They've been doing Red List assessments for some time. And so countries are reporting the number of threatened ecosystems per risk category, per ecosystem functional group. And you can see how that does change through time. So even in this short time between South Africa's assessments, we saw a real decline or increase in risk for coastal shrublands and grasslands, which, like a lot of coastal ecosystems, are threatened by things like development and invasive species. And that's an example of one of these threatened ecosystems, the Juneveld in Alexander Bay. Another one of the headline indicators is, stems from ecosystem accounts or natural capital accounts using the System Environmental Economic Accounting ecosystem accounts, which is the UN's Statistics Division's global standard for ecosystem accounting. And actually, from your national ecosystem accounts, you can derive two indicators: extent of natural ecosystems, A2, and also B1 services provided by ecosystems. And Australia's just completed its first set of national accounts, so is in a good position to do this. I've also just highlighted there's also condition accounts, which are not used in this because they're not very often done, and so I'll come back to that later. So how's the data availability for these indicators? It's not perfect. These were selected as headline indicators because of their potential, I think, rather than currently available data. So in the top panel, we've got, I think I've got a pointer, let's see, yep. That's where Red List of Ecosystems assessments are available. The red countries, like South Africa, they've assessed all their ecosystems more than once. So they've got a comprehensive data set to run with. Other countries, like these ones in Latin America, only the forests have been assessed. From those assessments, we can derive Red List indices for these countries if they have enough data of the right type. And down here we've got the where ecosystem accounts have been done, and we can see it's pretty similar patterns of white, like gaps of lack of knowledge, and I think this really is representative of places that have not potentially had historical investment in ecosystem data or are less inclined to take an ecosystem approach. You will, of course, notice that Australia is, we've just done our first accounts, but we've got lots of individual ecosystem assessments, which speaks to David Keith's hard work, and many of his colleagues and friends around him. But we haven't got a national Red List of Ecosystems assessment or even a state one. And another potential new initiative is this Global Ecosystems Atlas, which is aiming to develop a globally comprehensive ecosystem map product. And that is being led, the science of it's being led out of JCU, actually, Nick Murray, which is really exciting to see more Australian science leading the world. Okay, so just to summarise, we have this foundational data that we really need to support not just the GBF, but all these other products in between. And so both the ecosystem accounts and the Red List of Ecosystems, which underpin these headline indicators, require an ecosystem typology and descriptions, associated spatial data or maps, and indicators of ecosystem integrity, how these systems are changing. And these frameworks ask them that they are ecosystem-specific measures that really get to the characteristics of those ecosystems. But we also have these other indicators that are derived from these other knowledge standards that are derived that are important for the GBF and other policy settings as well that use the same information. So this includes the key biodiversity areas, which is another IUCN standard, and these new emerging private sector frameworks, which are, I think, running ahead of potentially where the science is at the moment or where the data compilation is, but has quite a serious potential to change the way that biodiversity is managed on private lands in a way that is worrying if we don't have the right settings and tools to enable that to be done well. And so they, for example, rely on maps and descriptions of ecosystems. They want to know the risk status. Are they threatened? So that's Red List of Ecosystems. They're interested in key biodiversity areas. So all these same data, and they want to know the condition of the sites. And so all these same data could potentially support this, which really talks to the investment needed. I've talked mostly about indicators and the role of this ecosystem data and knowledge in reporting on the GBF, but it's also really key to implementation of the GBF targets and goals. We did an analysis of the role of this information across the different targets and goals. So, for example, ecosystem maps, data on condition or integrity, and ecosystem descriptions are really key to implementing a whole range of the different targets. For example, it's very hard to know what you need to restore unless you have good maps and descriptions of those ecosystems to do effective restoration and that you're doing the right thing in the right place. The risk categories, the evidence has shown, for example, in South Africa and Colombia, that this is really critical to helping support priorities and threat diagnosis. I told before the example in Colombia, where they established protected areas once they'd done their Red List of Ecosystems assessment and identified just how threatened their dry forests were. This information can also help prioritise where to restore, which ecosystems are most at need of bolstering, I guess, or reducing risk or risk mitigation strategies, but also might help you monitor or try to mitigate the impacts of climate change or pollution. And then finally, by using these consistent definitions and standards, it actually has this great capacity for sharing knowledge and learning across different countries and institutions as well. And so this is an example of a workshop in Kenya, which was when they were starting their Red List of Ecosystems for coral reef ecosystems in the Western Indian Ocean. And they were holding a workshop that brought together experts on the Red List of Ecosystems and coral systems. And that included, down here, Luis Odunga, who led the kelp forest assessments of South Africa. And he came along to share his knowledge of doing Red List of Ecosystems assessments in marine ecosystems, and also included scientists from Malawi who were embarking on their own national Red List of Ecosystems assessments. So by using these common frameworks, there's much greater capacity for working together across countries and sharing knowledge. But South Africa is a particularly strong leader in that across Africa. So what's happening in Australia? So Australia's developed an NBSAP, which is this very nice document. We've signed the agreement. NBSAP is the National Biodiversity Strategy and Action Plan that the countries submit to show how they're going to implement their own targets and goals and indicators that align with the GBF. We have signed on, obviously, to the CBD and the GBF, but I'd say we're only tackling some of the targets and goals, not the whole, when they're really intended to be tackled as a whole to address the loss of biodiversity. So we have listed 7 action targets in there, and 6 of the enabling targets about mainstreaming, for example, but only partially. So it's a little bit cherry picking potentially, the bits that we are already doing, for example, or doing well in, or that align with national priorities rather than the whole. So, for example, protected areas, we're doing pretty well on the protected area statistic of, getting close to 30%, like you'll see in a moment. But then all the other clauses around participatory management effectiveness, potentially are not being measured or addressed as effectively. We have, in this document, we did not commit to using the headline indicator, so informally it looks like we're likely to. But we do, surprisingly, have low data availability and reporting capacity. Malawi and is more likely to be able to report on its ecosystem indicators than we are, and one of the poorest countries in the world. So these headline indicators that sort of relate to, just headline indicators more broadly. Red list of ecosystems. We have patchy, we've got 25, 30 assessments across Australia, which you can see up there. Plus we have a similar way of listing threatened ecological communities. We've got about 100 of those, but they're a bit patchy, and it's not a comprehensive assessment that can enable us to report on the trajectory of our ecosystems. So that's not really. Extent of natural ecosystems, because we just publish the accounts, we have a sort of a preliminary national ecosystem map that's aligned with global ecosystem typology that was done on the back of the accounts. And then that's being further worked on by David Keith over the coming years with an industry laureate. We're pretty good on our species data. We've got comprehensive red list assessments for our vertebrates, but we're really missing for plants. I think fewer than 40% of Australian plant species have been assessed. So I've given that an OK tick, blue tick rather than a green tick. We do have other complementary indicators such as the Threatened Species Index, which is a Tern indicator. We're doing pretty well on protected areas in terms of the area, we're not measuring so much the other aspects, and we're getting close to hitting the targets. We collect data on that. Rate of invasive alien species establishment is another indicator that's from Australia. We think the data are there, but it's at the moment not all compiled in one spot, ready to go. When we look at some of these foundational data that are needed, we're doing quite well on the accounts. We had our first set of accounts, so there are some gaps there I'll come back to. With the red list of ecosystems, that's the agreed protocol under the Common Assessment Method in principle for all states and federal governments to use for listing threatened ecological communities, but at the moment it's only being implemented in 3 of the states, so we'll see if that gets picked up, particularly federally. Doesn't want to go back. Australia's really investing in the private sector frameworks and is very interested in those. So it'll be really interesting to see how that develops, and I think that's going to be a key area as we move forward. We're doing pretty well, bit of a late starter, but we're getting there on the national ecosystem mapping. David Keith, as I said, just got a big grant to try and work across the states and federally to align with the global ecosystem typology and really try and bring together the data across the states. So it's a sort of upgrade on NVIS, which was developed for a different purpose. So that's underway, and we should have something by the mid to late next year. But one of the really key gaps, which is that box in green, which is these ecosystem-specific indicators of ecosystem integrity, which are needed for accounting, red list of ecosystems and these other things, that remains a gap. We've got a good set of what I would call sort of ecosystem agnostic or more general condition indicators in Australia, but we lack these ecosystem-specific ones that are required for those purposes with accounting and red list of ecosystems. What do I mean by that? Well, this is where we, for each ecosystem, we're picking variables that are really relevant to the ecosystem characteristics rather than general ones across multiple ecosystems. And they're also then tailored to particular ecosystem groups by turning them into meaningful normative indicators. So where going up means good and going down means bad. So we can interpret them easily. The global ecosystem typology can help do this because you can potentially look at groups of ecosystems and use similar indicators. So for example, we found with the coral reefs, we can transfer the same set of indicators and use them around the world in different coral reef ecosystems. So that's been one proof of concept. It's a research question, is how generalised can you be to still achieve this ecosystem-specific needs. Just to give an example, this is from the National Ecosystem Accounts, which are a great achievement given the time frame that they had to do them. And some of the key statistics that were published with that, many of them are really mean something. So, for example, or are useful or easy to interpret information. So, for example, that desert ecosystems make up half of Australia. That's really interesting. We can know what that means. We can see that 4% of natural environments adjacent to rivers was converted. That's meaningful and concerning. So obviously not doing a very good job of protecting our floodplains and riparian areas. And feral animals and weeds continue to spread. That's also something that we know is not good and that we need to do something about. The one that I've highlighted in red there is that half of Australia's mangroves had at least 80% canopy cover. Now, I'm not sure, I'm not a very good ecologist, but I'm an ecologist. I'm not quite sure how to interpret that. Is that good or bad? Is that normal for mangroves? I'm not sure. Is that the natural range of variation? And similarly, we reported NDVI for different ecosystem groups. And the key thing here, so that these are all different ecosystem functional groups from the typology, and the 3 different coloured lines are different years. So we can see that different ecosystem groups had different NDVI levels, index levels. It bounced around each year, but it's a little bit hard to know if that bouncing around is just stochasticity, like wetter, drier years. The first year was at the back of the drought. Is getting bigger good or bad? So I would assume for tropical rainforests, it's good to be more productive. So going up is a good thing. But if you're a grassland, increase in NDVI might actually mean more invasive species, for example. So it's, going up might be bad. And so we need to be able to turn these into indicators that relate to each ecosystem, and so we know what is within the range of natural variation and which directions are not good for that ecosystem. And then there's others for which this might not be a very meaningful indicator. So, for example, sandy shorelines, sort of in the middle there, I don't know, what does that mean? There's more seaweed on the beach the day that was measured? I don't know. It's hard. We probably wouldn't bother having that indicator in there for that system. So that's the sort of tailoring that I'm talking about. Tern can really contribute to this. And I think that what we'll probably need to do is have a think about how we can use this information that Tern collects in its many indicators and many data sets to contribute to this tailoring for the specific purposes of things like accounting and red list of ecosystems. Of course, these data sets serve many other purposes as well. So it's not hijacking them to turn them to one purpose, but understanding how they can help. So, for example, we've got these data collection standards, these standardised data that have been collected, compiled and aggregated, and that's a really useful tool. We have these very detailed plots, so we've got the sort of Oz plots over, 900 sites or something, and then a few sites with very detailed information that might be able to tell us about reference levels or normal range of variation for certain indicators, and then whether there are any things that are fluctuating or there's a directional change that we need to be concerned about and how representative they are. But the question is, how well aligned are these now with our the global ecosystem typology, and are they representative of those? So just these sort of analyses and understanding. And then we have these broad-scale data, remotely sensed data and soils and so forth, which would be really important for modelling and mapping and bring those together within that context. So there's a lot of work that I think that we can adapt to bring into to bear on this problem. Yeah. And just finally, just some challenges that I think we have globally, but also that require some thought, and also particularly in Australia. So one of the big ones is bringing together knowledge systems about ecosystems to classify, define and map ecosystems and measure their change. So this includes in particular bringing together Indigenous knowledges into this challenge. We've started embarking on that. Alice Young is a PhD student on mine. We've just started to touch and work on the Tiwi Islands of working with traditional owners on how they differentiate different ecosystems, what their names are for those ecosystems, and going on country and understanding some of the values of those ecosystems. That's just the first dipping the toe in, because that's a long-term process, and then understanding how we monitor the state of those ecosystems and how they're changing. But that's enabled us to map the ecosystems of the Tiwi Islands in language, for example. How we use this information is a universal challenge, and how we use it best, particularly to guide, I think, private sector action as well as government actions and priorities. We also have the problem that you alluded to before of this cross-jurisdictional challenge in Australia, where each state is doing things slightly differently with different objectives, and it just means we don't necessarily have this sort of coherent, consistent approaches. Everyone doesn't have to do the same thing, but it's just understanding how they relate and how those data can be used together, and how tweaks could be made that would enable them to be used together, I think, which is the big challenge. I've got a grant in on that, so fingers crossed it gets up, because that would be a really fun and interesting bit of work to do. So just to conclude, ecosystems are really central to achieving the vision and the goals of the GBF in Australia and globally. And that this data, this knowledge and the indicators that come from them can help us meaningfully monitor progress and implement the action that's needed to achieve the goals. But we've got, in Australia in particular, but also globally, this really high need for coordinated data and training and collaboration, and also strategic investment in data. We'll never have enough money to get all the information that we need, but how we can use that very strategically and bring together existing knowledge and data and experts. And then this last thing that just sort of weighs on my mind is how we can better use this information as we bring this new data together and this new knowledge for better outcomes for biodiversity on private lands. Thank you. I just wanted to also thank the many people who have been part of this work. Thanks.
Thanks, Emily.
Sorry, I have my emerge. Oh, sorry.
You're good.
There we go.
Yes. Emily's going to be back with us at the panel session after morning tea. So if you've got questions and you've got lots of time to think them up and discuss them amongst each other, and then hit it during that panel session. And talking of questions, we've got a little problem with Q&A. So we'd set it up so that everyone asks their questions through the Q&A app rather than us passing around microphones. But we recognise that not everyone can get to the app at the moment. And so I thought perhaps if you can't get into the app to find the Q&A, perhaps you could email your question through to me, and you'll need to remember that my email is beryl.morris@uq.edu.au, and I hope you found the wireless system, which is the State Library wireless free. So we are now into our biocomplexity section, and I'm going to be handing over the running of the symposium to Mike Grundy, and I'll just tell you a bit about Mike Grundy, because he is a long-term person in Tern. We're very honoured to have him here to run the biocomplexity session. He's an adjunct professor in the School of Agriculture at the University of Sydney, a member of the Wentworth Group of Concerned Scientists, a specialist advisor to Tern, chair of the Independent Science Committee for Accounting for Nature, and he did retire from CSIRO as Research Director of the Soil and Landscapes Group. And Mike has had a long-standing research interest in spatial soil and landscape science, and its application to agricultural and forest production, environmental protection, and system approaches to complex problems. And he has a very long bio, which I think you should be able to hunt down in our apps. But it's really great that he's managed to make it in here today, because he has been incredibly busy with his new hobby, which is looking and being a proud grandfather of 3 grandchildren, which is becoming an obsession to him. So thanks, Mike, for giving that up for us. I'll leave you to run the session.
Thank you, Beryl. Well, you won't hear much of me. I'm just standing up here in front and telling you what to do and when to do it. Looking after 3 grandchildren is not a simple process, so you can't use the KISS principle, and the same thing applies to our natural environment. So we're not starting with an exercise in simplicity, we're starting with an exercise in complexity. And those of you involved in systems thinking will know that is, you actually have to understand that you're dealing with complex systems. You're dealing with systems where a simple modeller's understanding of it doesn't work. Some of my modelling colleagues and I have had arguments about this over many years. You're dealing with systems where there are interrelationships numerous and difficult to understand, where there are major feedback processes happening, and where the end result can only be understood by the way in which we interact with them, because we're part of that system. So we thought we'd start with something difficult for you to get your head around with, and then we'll go from there. We're not, the speakers that we have in front of us are not giving us the final solution to these complex questions. That's good, because that's impossible, but what they are giving us is the various ways in which Tern is either helping us to probe and understand the functions, resilience, the interrelationships of these systems, or the emerging properties that come out of these systems, and so that we can understand where they're up to and where they're going. With that introduction, remember that your questions have to either go through the app or through Beryl, and we will now start with our first speaker, who is Irene Martin-Fores, and I'll leave her to introduce the talk to you.
Good morning, everyone. All right. I am going to talk today about tracking invaders, an invasive species index to revolutionize biosecurity. First, I would like to start by acknowledging the traditional custodians of the land. Okay. There are many threats currently impacting biodiversity. Among the most well-known ones within society, we can find pollution, climate change, land use change, and finally, biological invasions, or the introduction of species facilitated by human actions. Biological invasions is a lot more complex than we might think to start with. There are 3 main drivers of biological invasions that are predicted to significantly affect future impacts of alien species. In order of importance, there are trade and transport, climate change, and socioeconomic development. All 3 of them are expected to increase over the decades, and that is so that established alien species are expected to increase by 36% from 2005 to 2050, even when we consider the best-case scenarios. Globally, biological invasions are as costly as many natural hazards. In fact, they take the 2nd spot in the list, only after the damage caused by storms. For you to have an idea, biological invasions are 774% more costly than wildfires and 8% more costly than the damage caused by floodings. In the case of Australia, weeds and pests cost our country around 25 billion a year, and this is expected to get worse. Between 1970 and 2017, Australia spent or incurred losses totaling at least $390 billion due to invasive species. And globally, the cost was reported at 1.7 trillion. We cannot forget that there are countries that are not at the forefront of invasion monitoring that have rapidly increasing costs. I apologize for the presentation being like this because it didn't have any problem in my computer. So, biological invasions are by far the main threat to Australian threatened taxa. In fact, 4 in every 5 Australian threatened species are at risk because of invaders. And invasive species have already been involved in the extinction of at least 45 unique Australian species. This is the invasive species action curve. So at the beginning, a species is introduced, and then there is a time lag until the species can be detected in the wild. And during this first period of time, eradication would actually be quite feasible. Then the species starts spreading, and public starts becoming aware of this becoming a problem. But by that time, eradication is already difficult. And by the time we are actually able to develop biosecurity actions, eradication is unlikely, and only local mitigation is possible with increased cost and long-term management. So these would be the biosecurity actions depending on where on the curve we are acting. So before a species is introduced, we can focus on prevention, in early stages in eradication. At some point, it's only containment, or if we leave it too long, long-term management and mitigations. Focusing on the right side of the curve doesn't work. This is what we are currently doing, and less than 5% of all the established naturalized plant species in Australia are currently declared. So we don't have the capability of the capacity to leave it that long. Instead, what we need to do is be smart in the way that we are monitoring and focus on the early stages of the curve. So the most efficient monitoring times for invasive species is when the problem is not so obvious. So we need to invest on prevention and eradication by improving our monitoring techniques. Aligning with this, the Australian Academy of Sciences highlighted the need for a unified national biosecurity data system, and that the timely and accurate identification of invasive species is critical for environmental protection. So how can we do this? How can we contribute? In turn, we aspire to design an invasive species index. For that, first, we would like to focus on reaching a terminology consensus. Why is this important? This is an image that I designed to illustrate what's currently happening in the field of invasion science. So much like the myth of the Tower of Babel, where humanity was unable to complete the construction of a tower to reach the heavens because of speaking different languages, invasion science is built on different terminologies and frameworks leading to confusion and miscommunication. And this is a very oversimplified image. In fact, we have 64 different terms that are used to describe or classify non-native species. And what is more worrying is that they are often used indistinctly. Let's go with the most well-known one, invasive. Do we even know what invasive means? There are 2 different schools. One that defines invasive based on impact, on negative impact, and the other one that defines invasive based on spread. There have been attempts to actually merge both of them, but so far we are still lacking a consensus on what invasive means. And invasive is not a binary concept, it's a multidimensional one. So for this first step, we want to reach a consensus in terminology to provide a strong foundation to refer to biological invasions. For that, we have designed a survey to identify the preferred terms used by different stakeholders following a bottom-up approach. I am very happy today that I can actually give you the QR code to access the survey. So if you want to contribute to solve this complex problem, please go ahead. And you can also find me in the coffee break, which I'll give you some flyers with the QR code as well. So this survey will allow us to consider spread versus impact, definitions of invasive, avoid loaded terminologies, and hopefully will allow effective communication and management actions across different stakeholders. The second step is reaching an impact assessment consensus. Impact is also tricky. It's a multidimensional concept. There are 3 different dimensions of impact: ecological, social, and economic, with different frameworks targeting each of them or the overlap between pairs of them. But there is no one single framework that actually unites and integrates the information on all of them. And we don't know what we don't know. So a non-native species might seem harmless at the beginning, but novel conditions can trigger for it to become all of a sudden very problematic and have major impacts. So we cannot assess change based on today. We need to predict what's going to happen in the future, track how abundant species are, how far and fast they spread, and take this as an early warning, and consider the context. So strategies based on good ecological understanding and across ecosystems will give us a stronger way to monitor invasions. So, for that, we need to propose an integrative impact assessment technique. So consider negative and positive impacts, both of them, environmental, social, and economic ones, and consider context specificity and scale dependency for accurate assessments. The third step is design a methodological approach, including spread and abundance. Spread combines both spatial and temporal dimensions, so even spread can be ambiguous without clear and quantifiable criteria. And I wonder, when is spread significant enough for a species to be considered invasive? Well, that also is going to depend. It's not about just how far or how fast a species moves. It is about context. So the same species can move completely different distances in different places or ecosystems or situations. And what is significant depends as well on the taxa. We cannot fix just like a certain distance as significant. We cannot compare, for example, an invertebrate with a bird. Besides, natural catastrophes can boost spread in unexpected ways, and to complicate this further, spread is difficult to detect and we do not track it evenly. There's a lot of what we know is patchy and biased. So defining invasion by spread is less about ticking boxes and more about reading the full story in the ecosystem. And abundance, the big gap in reliable information to track invasions. Abundance patterns are spatially and temporally dynamic and do not necessarily correlate with spread and impact. So this complicates things even further. Inconsistent monitoring protocols and metrics across regions hinder comparability and synthesis of abundance data nationally and globally. Surveillance efforts often prioritize high-profile or regulated species, resulting in taxonomic and geographic biases. And citizen science, even though it provides a very valuable information and large-scale data, is often prone to biases such as over-reporting problematic species, misidentifications, duplicate records, and spatial clustering around urban areas. So for that, we would like to develop a robust and accurate methodology incorporating the biocomplexity of invasions. So integrate all of these dimensions, be suitable for early stages, ensure it's comprehensive, and have standardized guidelines. And combine rapid monitoring with field surveys, modeling, and citizen science initiatives. And finally, predictions, tests, and implementations. So forecast future trends under different land use and climate change scenarios, ensure that it aligns with the Threatened Species Index for biosecurity and conservation actions to be supporting each other. implemented at a multi-scale level, so not just species, but also populations, communities, and ecosystems, and start small and then improve it. So what would this look like? Because it's obviously a lot and it's very complex. So I've just designed a little image of what it will look like in my mind just for a single species, for buffalo grass. So we could have something like this. So the different axes are the different dimensions that we need to assess, and the polygon is actually the impact that the species would have. Thank you very much, and you can find me or ask me any questions later.
Thank you, Irene. We will have a panel discussion at the end of this when all of the speakers will be up there, and you'll have an opportunity to present your questions either through the app or through Beryl. Our next speaker is online, and so if you can either virtually or actually welcome Vilma Perez, please.
Good morning, everyone. Today I'll talk about the changes in environmental microbes in response to fire. But first, I just want to acknowledge the Ghana people as the traditional custodians of the Adelaide region where the Australian Centre for Ancient DNA is located. I also want to acknowledge that in ACAD we live and work on many different nations' land, so I want to pay my respect to the elders past and present. So environmental disturbances such as bushfires are becoming increasingly more frequent and severe due to the climate change and human activities impacting biodiversity and ecosystems, especially in regions like Australia. But to effectively manage and adapt to these changes now and in the future, we need to establish baseline of climatic and environmental conditions to understand how ecosystems have historically responded to these disturbances. And microorganisms can actually help us collect this type of information, acting as bioindicators. This is because they have a fast and sensitive response to changes in the environment, and also because they perform essential ecological functions such as nutrient cycling, organic matter decomposition, and plant growth promotion. So they're really the foundation of long-term ecosystem health and resilience, and the trajectory of ecosystem recovery is strongly linked to how microbial species and their function shifts over time. But we still don't fully understand what happens to these microorganisms over long periods of time after environmental changes. So how do we fill this gap? One of the tools that can help us obtain this information is sedimentary ancient DNA, which is essentially ancient environmental DNA from past organisms that have been preserved in sediments. Sediment accumulate over time in different layers, creating a natural archive that can help us study ecosystem changes from decades to thousands of years. So for example, in aquatic environments such as lake, like in this figure, the sedimentary DNA record captures genetic materials from organisms from the surrounding landscape and catchment, including soil, and from those that live within the lake in the water column and the sediment itself. So the DNA found in sediment can be a mixture of historical and ancient DNA molecules from different past organisms, from microbes to plants and animals. And we can also find modern DNA molecules from viable and dormant cells, especially from microbes living in the sediment. So although the information that each sediment layer provides can help us reconstruct ecosystems over time, this data is complex, and we also lack reference genomic databases for most of the species, including microorganisms, preventing the identification of them in the genomic record. So to improve this, we wanted to use environmental DNA recovered from modern soil samples from places near the sites where we collected the sediment record. This will help us reconstruct genomes from soil microbes organisms that then we can use to better identify soil microbes in the complex sedimentary record and also use them as reference to recover potential ancient soil microbial genomes. All of this with the aim of identifying ecological changes and genomic changes in microbial communities in response to fire over time. We are applying this method across different sites in southern Eastern Australia, but today I'll present the results from our study in Kangaroo Island, where we used sediment samples from Lashmar Lagoon and also soil samples we collected from the Tern collection on Kangaroo Island that have been sampled in 2018 pre-bushfire and 2020 post-bushfire. So the data I'm presenting today is part of a larger study led by Dr. Jonathan Tayla at the University of Adelaide, where the project goal is to improve reconstruction of ecological responses to past wildfire and use this information to build better predictive models for future climate change impacts. Our study site, Lashmar Lagoon, is located in Kangaroo Island in South Australia and was chosen because it provided fast accumulated Holocene lake sediment of this area. This is where John and his team collected a sediment core covering roughly the last 7,000 years and applied traditional paleoecological proxies like geochronology, pollen, and geochemistry to study climate and fire variability over time. And to complement these tools, we wanted to apply molecular techniques, in this case sedimentary ancient DNA, to study the biological adaptation of this ecosystem to change in environmental conditions by identifying genomic changes in microbial communities in response to environmental disturbance over the last 7,000 years. For this project, we performed all the lab work in an ultra-clean ancient DNA facility and followed strict ancient DNA protocols to prevent contamination from exogenous DNA sources. And to characterize the microbial changes in Lashmar Lagoon, we apply shotgun metagenomics, where we sequence all the DNA that has been recovered from the samples, and we use these short DNA fragments to identify the species present in the sample. We first investigated what type of organisms we had in our samples and how they change with depth and time. Here we have 3 bar plots with the taxonomic composition for archaea, bacteria, and eukaryotes domains at family level for the most abundant families in the sediment core from Lashmar Lagoon. On the left y-axis, we see the depth for each one of those sediment samples, and on the right, the corresponding ages for those layers, covering roughly the last 7,000 years. We notice a high diversity across the 3 domains of life, and as expected, a large percentage of the reads assigned to prokaryotes could only be assigned to archaean in blue and bacterium in gray, indicating the presence of novel microorganisms that have not been described. On the other hand, in the eukaryotes, species belonging to green plants dominated the samples across all samples, but for this presentation, I'll focus on prokaryotic communities. We also notice that overall, the proportion of the DNA molecules with characteristic signals of ancient DNA increased with the depth and age of the samples. We also notice a significant change in the taxonomic composition between 700 and 2500 years ago in the 3 domains of life. These changes seem to be driven by a sudden increase of halophilic families that contain species commonly found in saline and hypersaline environments. For example, in the archaea domain, we identify haloarchaea in yellowish, in bacteria, Halobacterium in yellow, and Dunaliella salina in eukaryotes in orange. The increased abundance of these halophilic and halotolerant species in our profile correlated with high calcium levels measured in the geochemical analysis done by our colleagues in the sediment record, which included carbonate and evaporitic minerals, suggesting high rates of salinity in the lake due to evaporation in this period, indicated that around 2500 years ago, a dry period started in this area. Also, previously Clark in 1983 reconstructed local fire history in Larchmont Lagoon using micro-charcoal records and found an increase in micro-charcoal around the same period, indicating high-intensity fires or more frequent fires in this region. So our finding and previously published work suggested a change to a warmer and drier climate in this region, and although these results have provided insight into changes in the past biodiversity of the lake, we were missing more information regarding the potential functions of these communities. But for this, we needed to reconstruct microbial genomes. This is because the functional annotation or the recovery of metabolic pathways from short reads, as we did it for the identification of the different species, is more challenging and inaccurate. So for this, we first recover microbial genomes from the soil samples of the Tern collection, and we call these genomes metagenome assembled genomes, as they were recovered from metagenomic data. So what you see here are all the species that we could recover some partial microbial genomes that we then use as genomic references to recover microbial genomes from the sediment samples. And here you can see all the genomes that we recover from the sediment samples. So we have each sample along the sediment core from Larchmont Lagoon and the number of metagenome assembled genomes, or MAGs, for each one of them. In total, 71 MAGs were recovered, with 67 of them showing ancient DNA damage that can correspond to genomes from past microbial species. Finally, we reconstructed the functional profiles for the 67 damaged MAGs along the sediment core. And although some metabolic pathway couldn't be fully reconstructed, we got some insight into their metabolic potential. For example, at deeper layers, we observed a higher abundance of carbon fixation and methanogenesis genes, correlating with periods of high organic carbon and low carbonates found by geochemical analysis to the sediment cores. That supported the previous finding of a relatively wet climate that promoted vegetation growth and reduced lake salinity. So these findings align with previous paleoenvironmental reconstructions, reinforcing our interpretation of climate and ecosystem changes over time. In summary, our results show vertical changes of microbial communities correlating to environmental change proxies. These communities show an authentic ancient DNA signal, and they likely responded to past environmental conditions and geochemical changes of the sediment during burial. These observations suggest that microbial communities are useful paleoindicators of environmental change over time. And we also show that by using modern soil samples, it allows to complement our sedimentary ancient DNA analysis. But more importantly, more optimization is needed for this type of ancient metagenomic data. Thank you for listening, and thanks to everyone involved in this research.
Now, both of those talks should be generating a number of questions. Are you all successfully getting your questions into the app or through to Beryl? Oh, I assume from that response that you certainly are. David Roberts is our next speaker.
Thanks very much. I'm joining you this morning from the Office of Nature Conservation in ACT Gov, and I want to talk about a couple of recent pieces of work that our team has done, pulling together some of our routine monitoring data to start looking at trends in condition of both threatened species and ecological communities across the ACT, including an adaptation of the TSX to the ACT context. So just for context or background to the talk today, the ACT Nature Conservation Act lists 65 threatened native species or threatened ecological communities, including the focus of today's talk, which are the yellow box Blakely's red gum woodlands, which are found right throughout the lowland areas of the ACT. And the map on the left-hand side here shows the potential extent of that threatened ecological community based on satellite data. And it is broken up into 3 different vegetation communities that together form that complex, including quite large areas of derived grassland in pink, which have had that native canopy removed. Now, the ACT is currently in the process of not just mapping the extent of that ecological community, but also its condition, using a categorisation scheme that speaks directly to eligibility of those remnants for listing under the EPBC Act. And you can see that categorisation scheme on the screen here. The main requirement for a remnant patch of woodland to be eligible under the EPBC Act is that the perennial vegetation cover is primarily native, and so that's that kind of top row of that classification scheme there. And then in areas of secondary grasslands where the canopy has been removed, there are some additional requirements that need to be met. So there needs to be evidence of regeneration of a eucalypt overstory, or there needs to be the ground cover species richness needs to meet particular forb richness benchmarks to be part of that threatened ecological community. And I'll come back and talk about this a little bit more in a couple of slides when I show you the results of some of the modelling work, because we've used this as a stratification to understand trends in different areas. Now, the ACT has a fairly rich and long-term data set of monitoring within this community over the past 16 years. So various different programs have measured or monitored some aspect of condition in the community at up to 500 monitoring plots each year, and you can see the location of those across the community on the plot here. Those data are collected for a variety of programs, but one of the key adaptive management programs that's really led to an increase in that monitoring effort over the past 7 or 8 years in particular is a management program that seeks to maintain intermediate herbage mass in these grassy ecosystems. So providing appropriate structural complexity and bare space to allow threatened flora and fauna species to persist in the landscape. So while these data sets have been collected for a variety of purposes, there's also a recognition, I think, that we now have a fairly rich data set at quite a high spatial and temporal resolution that allows us to ask some of those bigger questions around trends in condition across the landscape. So the approach that we've taken for this work over the last 6 to 12 months or so is to try and synthesise different views of condition that come from different data sets into a composite condition index that we can model in space and in time. So we can pull in information from LiDAR sweeps, which give us a view of canopy and shrub cover in that community. We can also bring in data from that monitoring data on ground, which gives us views of things like bare ground cover, native grass cover and herbage mass. And also from a subset of the plots, more detailed floristics observations, which give us a view of native species richness at the plot level, and also a metric used in the ACT known as the floristics value score, which is related to both the identity, cover and rarity of species within floristics plots. But really the key process I wanted to focus on was that middle table there, which is that there are established benchmarks for each of these condition indicators, which represent idealised best remaining condition in remnants of this threatened ecological community. And so the process that we've kind of gone through is passing these raw observations through those benchmarks and generating a composite index that gives a view of how close on average each of these indicators are to those reference conditions. So just to show you some of the early results from that modelling work, on this slide what we're looking at here is plots that fall within that top category of the scheme on the right-hand side here, where that ground cover, that perennial ground cover, is primarily native. So meeting that first requirement for EPBC eligibility. And on the vertical axis here is that composite condition index. So on this scale, a score of 1 means that each of those individual indicators that I introduced on the previous slide are either meeting or exceeding benchmarks. And as we get further and further below that score of 1, we're getting further and further from that idealised condition. So probably the first thing to notice here is in those kind of dark blue and dark green areas, which represent our highest condition remnants of that community, the temporal trends suggest a maintenance of condition through time within that management context across the past 16 years. But if we have a look at the lighter blue and lighter green areas, and these represent kind of one step down on that categorisation scheme that I introduced earlier, we can see evidence of declining trends in those areas. So in the case of that light blue trend, which you can see on the left-hand side there, these are native woodlands that meet eligibility criteria for EPBC listing, but evidence of declining trends coming through in this composite view of condition for that community. And that is really consistent with the temporal trends that we see in other parts of the landscape as well, where the ground cover rather is primarily exotic. And so we're looking here at plots which aren't meeting any of those criteria for EPBC listing, with significant declines occurring in those areas as well. And so really you could think about this as, you know, potentially a tale of two management contexts. In areas with initially quite high condition in a relative sense, there's evidence of maintenance of condition, but in a management context where management might be aimed at either improving or restoring condition, limited evidence to suggest that those approaches are being effective at a landscape scale. And so what this kind of approach we think can do is help us to identify areas within the landscape where we can go and ask some of those more targeted management questions. Where are we seeing effective management that is resulting in improved or maintained condition? And what landscape context might we be seeing limited efficacy? The other thing I wanted to touch on just really quickly in my last few slides as well is to talk a little bit about some work that we've done within ACT related to the Threatened Species Index. And I'm going to assume a high level of understanding with this audience of the TSX and what it does and what it seeks to do. But just wanted to, I guess, make the point that within the ACT context, while at a national scale and for most jurisdictions, there is a fairly consistent picture of overall declining relative abundance of threatened species in these aggregated measures. When we go to the ACT scale, it's actually the only jurisdiction in that TSX dashboard that shows a predicted slight increase. Although I'm sure you can see there in the top left plot that there's quite large confidence intervals around those estimates. And one of the things that I think is really interesting from the ACT context, at least, in the context of these grassy ecosystems, some of the most important threatened species from our conservation perspective are reptiles and invertebrates, which aren't currently captured in the national TSX. So we have done a little bit of work. I'll just skip over this slide just for time. We have done a little bit of work over the past 12 months within the ACT just to draw together our time series data for species which we have sufficiently robust monitoring data to feed into the TSX, including, if you have a look on the top right there, coverage of reptile and invertebrate species and also fish, which are not currently included in that national index. And we've actually gone through a process of bringing that data flow kind of processing in-house, so using the available script and workflow to allow us to kind of recalculate a TSX for the ACT region that includes those taxonomic groups. And I think what's quite interesting is when we do throw those in, we actually get quite a different picture about those relative trends in relative abundance of threatened species when we include those other taxonomic groups. There is a little bit of work to be done here as well to pull in some more data on birds, which local ornithology groups are working with us just through some data sharing arrangements at the moment. So this is just a snapshot of this point in time, and there is probably another 10 or 12 species which we can put into the mix. But just to reiterate that point, when we have a look at trends by individual species groups, you can see that invertebrates and reptiles are really the two taxa that are trending most strongly through time. In this data set, and really potentially pulling through that shift in both the direction and magnitude of change predicted. And so I think there is also potentially a piece of work to be done there within a particular region, a particular management context, but with additional taxonomic groups to tease apart the relative influence of individual species, individual time series in driving those trends. So it's certainly not a finished product by any means, but it's a project that's, one step along the way to trying to reinterpret some of those trends in a regionally meaningful context. So I will finish there, but just in terms of next steps, the ACT Gov is currently in the process of developing a revised nature conservation strategy, and both of these approaches here have the potential to be adopted as headline indicators to help inform progress against specific objectives in that strategy. Continuing to work with some local non-government data custodians as well to increase the representation of birds in particular in that ACT TSX. And eventually, there's a vision that both of these indicators could be progressed into some public-facing dashboards as well. Thank you very much.
Thanks, David. Well, you have heard your first 3 biocomplexity speakers, but don't worry, there's more. There's more after morning tea. But it is imperative that you get back here by 10:35. So you may now leave. You can consider your questions that I've yet to see too many of in the app, and be back here at 10:35 when we conclude our biocomplexity session. Let us start the next session of biocomplexity, and we're going to start with Tayla. And afterwards, we will line them all up here and we'll go through your questions. So remember, add your questions.
Just that one. Hi everyone, my name's Tayla Lawrie and I'm the Project Manager for the Threatened Species Index here at Tern. And today I want to talk to you guys about tracking Australia's biocomplexity. So we already have our Threatened Species Index for Australia, but could we also have an Invasive Species Index? How might we make it, and what can we learn from the Threatened Species Index? Now, when I refer to the Invasive Species Index, I'm not referring to the 4-part plan that Irene described earlier, but I'm talking about a specific tool that we can use to track abundance of invasive species. So, for those who are unfamiliar, I want to quickly summarise what the Threatened Species Index is. So, it is a key biodiversity indicator for Australia. So, it's an objective and quantitative index that measures changes in the abundance of Australia's threatened and near-threatened species. So, the index itself is updated and published each year on our data visualisation tool, which is up on the slide here, and can be accessed via our website. It's based on data for over 24,000 monitoring time series that come from all across Australia. It covers 335 species of birds, mammals, plants and frogs, with reptiles coming soon. And it is based on the Living Planet Index methodology. So the Living Planet Index is a global index that looks at vertebrate abundance. Now it is transparent and scalable, so much so that we have taken it and applied it to threatened species in Australia. And to quickly summarise the results of the 2024 index, between 1985 and 2021, there was an average 73% decline in the relative abundance of Australia's threatened and near-threatened bird, mammal, plant and frog populations. Next up, I want to walk you through what I think an invasive species index might look like, based on what I know from the Threatened Species Index. This would look familiar. I've just taken the Threatened Species Index tool, changed the name and flipped the trend line. Obviously, I've made this up, but the point that I'm trying to make is that I think there would be a lot of similarities between the two indices. It could be an index that measures changes in the abundance of Australia's an abundance of Australia's invasive species. It could be a centralised repository of invasive species monitoring data. It could cover all major groups such as plants, animals, and even fungi and microbes. And it could be based on the Living Planet Index methodology if that was considered to be fit for purpose. Now, creating this index won't be simple. I think we'll need to bring in invasive species experts who understand the complexities of these species and combine that with what we know about the Threatened Species Index and our experience in creating this kind of tool and the challenges that we've had to overcome. Speaking of challenges, I want to walk you through some of the challenges we've faced in building the Threatened Species Index and how I think they might apply to an invasive species index. And by no means are these 4 things an exhaustive list. First up is nomenclature inconsistencies. So the Threatened Species Index brings together hundreds of different data sets from across Australia, And harmonizing those data sets can be really complicated if you have unresolved names, so by that I mean scientific Latin names, and if you have conflicting statuses, so how threatened a species is considered to be critically endangered, vulnerable, etc. To give you an example of some of the inconsistencies we see for threatened species in Australia, in Australia, we have a number of taxonomic databases that list all species known to occur in Australia. For our fauna, that is the Australian Faunal Directory. Take this one species, for example, the Norfolk Island parakeet. The name given to the species by the Australian Faunal Directory is Cyanoramphus cooki, and I'm sure I've butchered that, and I will continue to butcher all these names. But if we look at some of the conservation statuses applied to the species, we see some major inconsistencies. So first up is the EPBC Act. They actually use the same name as the Australian Faunal Directory, which is a great start considering they are from the same department. And they list the species as endangered. Next up is the 2020 Action Plan for Australian Birds. Now, this is a book produced by bird experts from across Australia and is considered a key source of information for birds and for bird conservation statuses. Now, according to the Action Plan, they actually considered the Norfolk Island parakeet to be a subspecies of the red-crowned parakeet, Cyanoramphus novanzelandiae, and it lists it as vulnerable. And I can keep going. So take the IUCN Red List, for example. Now, due to the nature of the Red List that often provides statuses at the species level, they actually don't recognise the Norfolk Island as either a subspecies or its own species, and so it kind of lumps it all under the red-crowned parakeet and lists it as least concerned. So now we've got 3 different authorities recognising 3 different names and applying 3 different statuses to the same species, and it's these kinds of inconsistencies that are a huge headache for us when we're trying to merge our data sets and build our trends. And we know that invasive species face similar challenges, as Irene mentioned earlier, particularly when it comes to defining what an invasive species is and the level of impact it's having. Next up is taxonomic and spatial data gaps. So collecting abundance data can be really difficult for specific species in particular environments, which means we often see taxonomic and spatial gaps in our national data sets. And this is certainly the case for the Threatened Species Index. In terms of spatial gaps, you can see on the map on the left there, which is a map of all monitoring sites included in the TSX, that there is a considerable bias towards the more populated areas of the country. Now, yes, this is where most threatened species occur, but there are still gaps even when that is considered. In terms of taxonomic gaps, the plot on the right there, which is a number of time series per taxonomic group, we can see that we have a lot more data for birds than we do for mammals or plants. These gaps are really driven by a number of factors, and this is in no way exhaustive list, but I think detectability, so is it easy to find and see? Public awareness and bias. I don't know anyone more obsessed with a particular group of species than people are with birds, so it's not ridiculous that we have a lot more data for birds than we do for our other groups. And location, how accessible or remote is the population we're trying to measure and monitor. And I think invasive species faces similar issues. So take this example of the cane turd versus the quincunx earthworm, which are 2 alien species in Australia. Now, detectability and public awareness is much higher for the cane turd than it is the earthworm. I certainly know that I had never heard of the earthworm when I was looking for this example. But these differences ultimately lead to probably higher monitoring for the cane toad and therefore probably more data for the cane toad in something like an invasive species index. Also contributing to some of these gaps we see in the Threatened Species Index is the methods being used to monitor. So we have our active monitoring, so that's direct human observation, boots on the ground, and then we have passive monitoring, so things like camera trapping and acoustics. The pie chart on my slide there shows that only around 1% of data contributed to the Threatened Species Index was collected using passive monitoring methods. Now, I don't actually think this is surprising. If we think back to the start of our data set in 1985 or even earlier, boots on the ground is probably how we did most things. But we know that technology is improving, and I think that there is a huge opportunity for us to better utilise the data collected using these passive monitoring methods to fill some of the gaps I just mentioned for both invasive and threatened species. Our next challenge is the siloed nature of monitoring data and monitoring and data management. So in Australia, environmental management and therefore monitoring is often primarily the responsibility of individual states and territories, or even specific natural resource management groups. Each jurisdiction has its own legislations and its own programs, leading to differences in what is being monitored, where and how. But we know that species don't care about our imaginary borders. And it's this kind of decentralized approach that makes accessing the data a huge nightmare for us, and also impacts how effectively we can monitor these species, both invasive and threatened. And our last challenge in creating an index is the hesitation to share data. So this is a huge thing for the Threatened Species Index. We often find that there is quite a bit of hesitation in sharing sensitive site location data for the fear of coordinate leaks and potential poaching risks. And for our academics, a lot of times there is hesitation simply because they have yet to publish their data. Now, these next 2 points are somewhat speculative, but I think they might be relevant to an invasive species index. And that is for private landholders, I think there might be some hesitancy to share data for the fear that specific control measures or land use restrictions might be placed upon them. And if they are already managing the invasive species on their land, they might be worried about the perceived ineffectiveness of the efforts and all the money that they're spending. It's hard to know how much these concerns will impact an invasive species index, but what we know from creating a threatened species index is that they're all manageable, and that is through communication on data handling and specifically data sharing agreements. I think we can learn a lot from the Threatened Species Index that would be relevant to an invasive species index, but I want to finish up this talk by discussing how I think we can and should move towards more linked national biodiversity indices. So if we can move beyond those kind of isolated trends and track invasive and threatened species together, and even bring in trends for common species and look up to the ecosystem level, I think we can have a more holistic view of ecosystem health. And it will allow us to answer really important questions like, are threatened species increasing where invasive species decline? Now, we know that there is localized evidence that this is true, say, for example, on Macquarie Island. But it would be really awesome if we could map this at a national scale. But when we're looking at these kinds of correlations, I think we also need to take into account the fact that the resolution and timing of these threats are really complicated. So we know that there is never just one threat that is impacting a threatened species, and we know that threats can be compounding and can take a while to impact a population. But I think if we can pick apart those things and we could possibly even use one trend as a proxy for the other, or even use that data to make predictions about the future trajectories of these species. To overcome some of these challenges I mentioned today, and to build an invasive species index and have a more holistic view of biocomplexity in Australia, it would be really awesome to see, and it's going to take a lot of work and a lot of collaboration, but I'm really excited about it. Thanks.
Thank you, Tayla. I'm sure you've got a number of questions that you've already entered into the app, so keep doing that. Our next speaker is Brendan.
Yep, thanks very much for inviting me to be here. So I'm going to be talking about the Blue Mountains Hub for Ecology and Conservation. I am working at the Hawkesbury Institute for the Environment at Western Sydney University, and this is a project that has been developed by Western Sydney University, Tern, in partnership with Adaphic Scientific, who are a scientific instrument company based in Melbourne. So the central goal of this project is to establish a permanent plot network with a focus on using innovative technologies that allow us to do real-time monitoring of ecological variables. And the intention is that we'll have this long-term data set that will then allow us to link to the global ecological monitoring community and also be of use for Earth observation for calibration and validation. So I think most people probably know where the Blue Mountains are, but for those of you who don't, it's just adjacent, just west of Sydney. So it's around 1 million hectares. So the Greater Blue Mountains World Heritage Area is around 1 million hectares of temperate eucalypt-dominated forest. So this is it's one of the largest tracts of protected bushland and forest in Australia. But it's also adjacent to one of the largest and most rapidly growing urban areas, and it's been subject to some really dramatic impacts from disturbance events, most notably the Black Summer bushfires in 2019 and 2020 and the drought that was associated with that. So the central aim of this network is to enhance ecosystem monitoring and biodiversity. And there are really three major things we want to do in terms of long-term monitoring here. The first is increase the number of inventory plots. So Australia doesn't have a national inventory plot network, a system where people go out and measure the number of trees, the size of those trees, and what species they are. And this is quite a big deficiency in terms of how we're able to monitor forest dynamics. So we really want to cover off on that. And this is particularly important in terms of, as we approach in the next decade, we're going to be looking at net zero targets for Australia out to 2035 and then further out to 2015. This is really going to bring a tsunami of change in Australian policy, and being able to have accurate estimates of natural carbon storage is very important. So that's kind of one of the big motivations. Another is to have real-time, very data-rich monitoring of ecosystem processes, and in this case, a lot of that is the physiological processes of the plants, which I'll describe in a bit more detail in a second. And thirdly is monitoring of biodiversity. And so one of the things that we're hoping to achieve, so I'm working very closely with people like Belinda Medlin and Ben Smith at the Hawkesbury Institute, who are process-based land surface modelers. And so these kinds of data sets are really essential in terms of improving ecological forecasting and predictions of ecological risk. So just in terms of where we are, we have 35 plots in a transect across the Blue Mountains. The base of it is actually the Cumberland Plains Tern supersite, which we manage at Hawkesbury. It goes up over the mountains and then into some of the drier sites in the central west. And we're trying to cover off on the environmental space. So the transect runs for about 165 k's. It's got a good elevational gradient that it covers. And if you look over, this is the Cumberland Plains here. And then as you go over the mid-mountains into up around Katoomba, you have a lot wetter, cooler environments, and then a lot drier in the forests and woodlands out in the central west there. So the plots are 30 metres in diameter. All of the trees over 10 centimetres DBH are measured and tagged. We have a larger plot of 200 metres overlaid over that for fauna monitoring, and we're covering all of the major vegetation types, dry sclerophyll, wet sclerophyll, and rainforest. We have all of this data now. It will be loaded into the Tern website as soon as we can arrange for that to happen. So we've got all of the individual tree data, details on the sites, and access to those sites. I think really the biggest ticket item here, though, is that 10 of the plots will be monitored for real-time monitoring for physiological processes and detection of fauna. So what we can see here is we have in selected trees in each of the plots, we're going to have sap flow sensors that will provide data on water use and transpiration, water content, microtensiometers, which measure water stress, dendrometers for growth. This will be paired with micromet, soil moisture, soil water potential data, and acoustic monitoring using, fauna monitoring using acoustic sensors, camera traps, light traps for insect diversity, phenocams to track phenology. And this will all be pushed to a telemetry hub, going to a dashboard. So this is from one of the demonstrations plots that we've had running for a couple of years in the lower mountains. You can see here every 15 minutes all of the data are pushed. It's easy to plot these on a dashboard. And so these can not only be used for scientific research purposes, but of course for community outreach, environmental education, and those such things. We've been testing some of the sensors at the Hawkesbury campus. So we have here readout for sap flow and some of the more novel sensors here. This is the microtensiometers that have recently been developed. So measuring water stress remotely in plants is historically has been very challenging. This is the first time we've put these sensors into eucalypts, and we're very happy with the results in terms of performance, reliability, and durability. We also have been very lucky to have Kim Calder's group out. He's running the Space Twin project out of Ghent University in Belgium. We've had 3 of his PhD students out who've just scanned all 35 of the plots. So we have TLS point clouds for this, and in the coming months we'll be working on segmenting and processing those data. They'll be complemented by aerial LiDAR. So this is the Zenmoose L2 system that we have. And we'll basically be scaling up from the manual measurements to TLS to larger areas scanned by ALS and then up to Earth observations. But none of this is really workable without having that ground truthing data. So, just to emphasize that that's a very key component. And just to finish up by saying we're very, very enthusiastic about collaboration, particularly within the Tern network. So please come and have a chat over lunch or over the coming days if you're interested in collaborating on any of these projects. And finishing just by thanking many of the people who've been involved in setting up the network and in the initial surveys. I can't show everyone here, but greatly indebted to all of the enthusiastic support that we've received. Thank you.
Thanks, Brendan. And now your final talk in this double barrel session is given by Zachary Amir. And after that, we will have a panel discussion out the front. So I still can't see any more questions on your app.
Hello, everybody. My name is Zachary Amir, and I am working with Tern to help develop the Wildlife Observatory of Australia, which is a national platform for wildlife camera data. First, I'd like to begin with an acknowledgement of country. We acknowledge the traditional owners of the lands on which we meet here, the Yagara and the Turrbal peoples. We also acknowledge the traditional owners of the lands on which we've traveled from and where we conduct our research. We acknowledge their continuing connections to their culture and pay our respects to elders past, present, and emerging. I'd also like to begin by acknowledging the WildObs team. This project is much larger than myself, and we've got many different partners such as QSIF, ARDC, UQ, Tern, and Bush Heritage Australia, all contributing to develop this platform. And to begin, I'd like to highlight an impact study. We worked with WWF to study the impacts of the 2019-2020 bushfires across Australia. WWF, in collaboration with Google, set up over 1,000 cameras across 9 landscapes where the Black Summer fires had burned, generating over 7.5 million images of wildlife. We led the analysis here, implementing single-season occupancy models that account for imperfect detection of species, and we assessed each species' response to various fire variables in addition to other spatial temporal covariates. We compiled a final report that included results for 26 species, and I've just pulled out the results for koalas here. We can see a slight moderate increase in occupancy through time, though noting a very large confidence interval at the beginning of our sampling. And fire was not the key driver of the species occupancy, instead it was recent rainfall patterns. If you would like to learn more about the Eyes on Recovery project, we have a QR code here where WWF has compiled a very nice report with lots of great figures and images. But we learned a few lessons from this that I'd like to highlight. There were major gaps in accessing pre-fire data. That data may have existed, but it was siloed and inaccessible. This revealed how limited data sharing can hinder coordinated responses to unprecedented events like major wildfires. When we were able to conduct these surveys using a somewhat standardized and scalable approach, there was a lot of value for generating continental insights. Ultimately, this highlights the importance of using data standards and developing sharing agreements to enhance future reusability. Ultimately, we need a national infrastructure to store, access, and analyze this sort of camera trapping data. Australia faces a few unique challenges. We're a leader in setting cameras across the nation, but much of this data stays siloed and isolated. We also lack resources for managing, analyzing, and sharing this camera trapping data. Many people are still manually reviewing lots of camera trapping images, storing these data offline and in hard drives that will get lost. And then finally, as many of the other speakers have already highlighted, we really need this data for conservation, management, and research. We can use this data to inform long-term trends like the Threatened Species Index or State of the Environment reporting. Therefore, we're proposing WildObs as a potential solution. Our goal is to revolutionize wildlife monitoring across Australia by providing scalable, integrated system that supports biodiversity, conservation, and research. So how is this going to work? Here are a few images from our prototype website still in design. We're working with AAF login credentials, and once we log in, you will see a static snapshot of our database, as shown here on the right, and then you will have access to see our various tools we're developing, such as our wildlife image management platform, tagged image repository, and our database of camera trapping records. And just to dig into each of those, let's go through an example of the WildObs workflow. Let's imagine we just came back from our camera trap survey and pulled the images out of our camera traps. Whether we're using an AI computer vision or manually reviewing the images, we somehow are converting these images into a spreadsheet. We've developed a data standardization process using R Markdown scripts that produce HTML documents describing how we've manipulated and transformed and standardized the data, which we then provide back to our data collaborators. Once the data is standardized, it can go to two places. The first is our camera trap database, where we have strong links with groups like the State of the Environment, Tern's EcoPlots, Atlas of Living Australia, EcoCommons, and the Threatened Species Index to ensure that our data is incorporated in routine monitoring. We're also developing a tagged image repository, which has been hosted by the Atlas of Living Australia. The idea behind this tagged image repository is to encourage the development of bespoke computer vision models. Once these models are developed, we can incorporate these into our wildlife image management platform, and you can select from multiple different computer vision models for the ecosystem or species of interest that you're working with. Ultimately, this feeds back into how we analyze our images, creating a closed positive feedback loop that can ideally develop lots of computer vision models to enhance the way we classify our images. I'd like to take a moment to highlight that our first data set has been published with the Atlas of Living Australia. If you would like to access images from, I believe it's 29,000 records, occurrence records from the Australian wet tropics. We can also use the Gala R or Python package to download these images directly to your computer, and they're all tied to occurrence records. So this is a very valuable resource, and we'll continue adding to this through time. Now, the Wildlife Image Management Platform is a pretty exciting tool, so I'll give a quick walkthrough. This begins where users upload wildlife camera data and select their data terms and how they would like their data shared or stored. We use a tool called MegaDetector, where we can classify images as to humans, blanks, vehicles, or wildlife. When we have those wildlife images, we can apply these computer vision models that I was just speaking about earlier. When we have anything less than 97% accuracy, or whatever number we're comfortable with, we can make sure to incorporate humans in the loop to review any suspect images. Once the data is annotated, it'll be exported into our camera trap database, and by using a very specific camera trapping data standard that maps onto broader data standards like the Darwin Core and Australian Biodiversity Information Systems, we can ensure that this data is usable by other parties as well. Just to demonstrate our growing data set, here is a map of Australia where we currently have camera trap data from our data providers. Obviously, there's a pretty large bias around urban centers in the coasts, and we're hopeful that we can begin filling in more of these gaps. And to describe some of the species we have in our data set, unsurprisingly, macropods and marsupials comprise more than half of the data set. But we also have a pretty large number of records for carnivores, which is namely like a lot of feral cats, foxes, and dingoes. And surprisingly, passerines or songbirds comprise almost 10% of our data set. And here is a graph of our number of detections through time, noting that it should be an exponential increase, but we're still facing these bottlenecks in processing images into spreadsheets. And to demonstrate how we can use this growing data set to answer ecological questions, I'd like to highlight a case study we're currently working on. This work is being led by Tom Bruce, who presented Wild Ops to the Tern Symposium 2 years ago. And we're asking the question, do dingoes suppress the abundance or behaviors of foxes and cats? On this map here, we have the distribution of cats, foxes, and dingoes across Australia, overlaid with our landscapes where we've sampled with camera traps. When we look at the number of landscapes with different combinations of cats, dingoes, and foxes, we can begin to compare cat populations in areas with and without foxes, so on and so forth. And given the wide spread of the data, we can also ask if this relationship varies across ecological gradients. So we're testing these results, or this analysis, across deserts, grasslands, and forest ecosystems. And today I'll just be highlighting the results from the forest ecosystems. So here I will show a graph describing animal activity patterns, where the y-axis denotes how active a species is, and the x-axis denotes what time of day, noting that midnight is centered in this graph. The green trend line will represent the dingo activity pattern, and the blue and red lines represent cat activity patterns, though the blue line is where cats do not co-occur with dingoes. Here we can see that the blue trend line is shifted lower in the midday and shifted higher around midnight. This is a significant effect as well, and this suggests that dingoes indeed push cats to be more nocturnal, but this result was only significant in forest ecosystems. Finally, when looking at the abundance of how dingoes affect the abundance of cats and foxes, we have dingo abundance on the x-axis and cat and fox abundance on the y-axis across all of our sampling locations. We saw no clear suppressive effect from dingoes suppressing cat abundance, and that same effect was found with foxes, same lack of effect. This suggests that there's no clear evidence that dingoes reduce cat or fox abundance within forest ecosystems, but it's important to note that this work is ongoing. We're still collecting data and applying various analyses to this data set. Finally, I'd like to thank our partners. This project would not be possible without many different organizations sharing their data and contributing funding, attention, and effort. And finally, if you have camera trap data or would like to use any of our tools, please reach out. We have a QR code that goes to our website, wildobs.org.au. And that's it for me. Thank you.
So can we have our talk speakers please to choose your seat? I'm sorry for taking so long to get that set up. It was taking a little while to respond. I have one question against that session. Against the first session, and the last time I looked, one against the second. In the absence of further, there's many other questions against the second. That's good. In the absence of any other votes, any other questions, I will take the chair's prerogative of asking the sorts of questions that occurred to me, but it may well be not necessary. Okay, from the first session, we have a question from Jill. and which I think probably could be answered by all of you, but the question is, what is the most important trend in biodiversity monitoring for the next 15 years that Tern might be able to support? Now, I've got a pretty good idea how each of you are going to answer that, but why don't we start with...
Well, oh, okay, there we go. Yeah, I'd probably say ecosystem state, in the sense that I think a lot of these things, things like invasive species and other pressures, all can. So you can measure each of those separately, or you can measure the response of ecosystems. And so we said it's not one thing; it's three things. It's ecosystem area, integrity, and risk. Are three key variables for understanding the state of biodiversity, because they can include things like composition and function in them, in sort of implicitly or explicitly. So I'd probably put that in. I think it can integrate all the other things that are really important, particularly ecosystem integrity, I think.
Okay. Brendan?
Thanks. Well, biodiversity isn't my core area of research, but I just wanted to make a quick point that I think covers across a lot of the monitoring technologies, which is one of the big barriers, and this came up in the last talk, is we have sometimes a huge amount of data collected, particularly by things like acoustic sensors, but also by LiDAR, and it's becoming very easy. It's becoming easier and easier to collect huge data sets, but it's still quite difficult to process them. So there's that barrier that is presented by a lack of accurate automation of analysing those data sets, and I think that's should be a big trend. I know that people are making progress in that area, but it's still surprisingly slow, considering everything we hear about AI and machine learning.
Thanks.
Yeah, definitely echo that last point. Probably also, I would say, the ability to generalise across contexts as well. So, being able to leverage data from different contexts and be able to start asking questions about the efficacy of management programs as well. So, are these things that we're measuring responding to the way that we're measuring them and managing them in different landscape contexts?
Thanks, Dan.
Thanks. Yeah, short answer, which trend? All of them. But I would say something that hasn't been mentioned today that I think is an important thing to think about is genetic diversity as part of biodiversity, as we haven't really got a good way to measure or track trajectories in that. So that's all I'll add.
Yeah, my response was going to be similar to Tayla's. I think everything is the answer. Like, even us, our biodiversity, are part of it. And there is no a simple answer for any of this. So any measures of any taxa, any trends are going to be useful. And it's about integrating them and being able to standardize the way we measure things and the way we can combine them so that it can be useful. And as Emily said, yes, if we can integrate this at the ecosystem level, that means that we are doing a really good job then.
Yeah, I think the fellow speakers here make really good points that I don't have that much to add on. I echo a lot of their sentiments here. I think the important thing to keep in mind with trends is, what are we interested in measuring? What's our specific response variable at this time? I think there probably will never be one trend to rule them all, but there will be definitely an opportunity for a synthesis center to bring together all these trends to enable better interpretation and applied interpretation for these trends to specific contexts.
Okay, thank you. Vilma, are you still with us? Perhaps not. Okay, there are a number of... We're not going to get through all of these questions, so we'll hand these questions on to the panellists later on. This is a question, I think, for Tayla, but possibly also for the indices we're talking about around invasive species as well. So for the trend data being ingested by Tern, do we have a strategy for not publicly exposing geolocations for threatened species? And the same thing applies for invasives.
Yes, so something with the threatened species indexes is when we publish all of our data that can be used by anyone, we actually de-identify the sensitive location data. So we actually zoom out and give a general location to the Ebro subregion. So we are protecting the location of those threatened species, and that is really important for all of our data providers to feel like they can provide their data and that it's safe.
I think for invasive species is not the similar situation, because we actually want to know where they are so that we can do something about it. So yeah, it is very important for threatened species index to have some precautionary measures around it, but for invasive species, I don't think it should be an issue.
Okay, the next one is a question for Alison. From Alison, for whom? Okay, so the question is, and this could apply again to all of you, but what plans have you got for sustaining your work over the long term? What level of minimum data might be acceptable if all the places can't be maintained? Sorry?
Oh, that's for me?
For
okay.
Brendan.
Yeah, I mean, well, sustaining long-term monitoring is always the number one challenge for these kinds of networks. So we've had good support from the university. Our hope is that Tern will continue to provide some support for the monitoring network, which is, attached to the Tern super site. But we also, I think, working with the university community within Sydney and also the New South Wales state government, we'll be able to draw on enough support to keep that going. The other ace up our sleeve we have is that around 95% of our permanent staff members, researchers in Hawkesbury Institute live in the Blue Mountains. So we have a huge amount of passion and support for continuation of that network into the longer term. But it's always a challenge.
Thanks, Brendan. And I do think that's actually a question for all of you, but I won't get you all to answer it. But the question always is, when you're dealing with something that's essentially about monitoring long-term processes, how on earth do you keep doing it? A Blue Mountains question, and I'm going to ask this one because it actually includes the soil. Is there any work being done to characterise subsurface structure deeper than soil, deeper than the shallow soil? And do you think it matters to understanding biodiversity?
Yeah, we don't have that as part of the plan at the moment, but that's definitely something which is important in terms of determining the structure of the communities above ground. We have a big team of soil microbial biologists who are more focused on the diversity of the microbial community, and they'll be coming in and measuring genetic diversity of that community. There's an interesting mix of substrates, largely sandstone, but we also have some basalt caps up there which really affect the community composition.
Yeah, you must have complex hydrological processes operating in the regolith that play through into your vegetation community.
Yes, so that's a really interesting point, and I think one of the problems with measuring things like soil moisture is that it's not representative at all a lot of the time with what the trees are experiencing in terms of where their roots are. And so I think one of the things that I'm hoping to get at, how water is moving through the deeper levels of the landscape is, how much stress are those trees being exposed to when you have a rainless period and when you have the surface soil layers drying out versus the access to deeper water in sandstone, for instance. Like this water percolates through the sandstone for a long time.
Thanks, Brendan. And perhaps I think we are out of time now. This is the end of this session. And I'd like you all to thank your panellists and your keynote speaker for today. And let's hope we get these questions working into the next session. And I'll hand out. And I think I'm handing over to Beryl.